Alright class, so in this video I'm going to show you a series of pictures and we're going to portray what the cell's going to look like in each stage of mitosis. Okay, we're actually going to begin by looking at our cell in interphase. So this is going to be our cell in interphase. We're going to have a nuclear membrane. Our chromosomes are going to be inside, so I'm going to draw our chromosomes inside, and instead of making them like dumbbell shaped, I'm going to do them just as lines, but two sets of chromosomes that'll look like that. Okay, so if we look at that cell from all the stuff we've done so far, what is that cell in terms of sets of chromosomes? Would this cell in interphase be 1N or 2N? It's a really good question. So remember, we're going by sets, right? 1N and 2N means how many sets do we have? So the way to do that on a picture like this is honestly to simply just ask yourself, how many purple chromosomes do we have? 2. How many orange chromosomes do we have? 2. So this cell is 2N. And then I want to show you what this is going to look like. We copy DNA, so we've done this. Copy our DNA, so now our chromosomes look like this, right? They're going to have sister chromatids. So they're going to look like that. We're still in interphase, right? This would be like the end of interphase. Hey, is this cell still 2N? Same question. Remember the sister chromatids that are present, those are 2 chromatids but still 1 chromosome. So 2 purples, 2 oranges. So yes, that cell is still 2N. Alright, so that's the cell we're starting with now. And we're going to show what it's going to look like as we go through mitosis, stages of mitosis. Right, so here's prophase. Okay, we've said, and this we're going to tie in what we saw with our previous video. So we said the nuclear membrane breaks down. So we can easily show that by just drawing like a dashed line now instead of a solid line. Our chromosomes would still be inside that. Right, we're just starting to break down that membrane around the nucleus. And then the last thing in here is we said we start to build the spindle. So the spindle early on is going to look like this. I usually just use like some asterisks at each side of the cell and then draw some lines that come out of it like that. Okay, that's going to be the spindle that we're building in these pictures. Alright, so that is prophase. Okay, we get down to prometaphase. So we said our spindle is finished forming. When the spindle's finished forming, it's going to look like this. So that's the spindle, fully formed spindle when we get into and finish prometaphase. We're not going to have a nuclear membrane anymore. And then these chromosomes are going to attach to our spindle. Right? So that, don't get too caught up on the technically how I'm showing you the chromosomes are attached. All that really matters for what we're doing is to recognize, oh yeah, those chromosomes are now attached to our spindle, like they're touching and lying on top of the spindle. Alright, so that is prometaphase. Okay, here is metaphase. Still going to have a fully formed spindle. And now these chromosomes, what we said about them is they're lined up. Right, so the chromosomes are going to be lined up in some plane of the cell that looks like that. Okay. We can turn this entire cell, we could rotate it and have the spindle going from the top down and the bottom up and draw our chromosomes going left to right. It's the same thing, they're just lining up in metaphase. Okay, and then we are separating sister chromatids in anaphase. Still have a fully formed spindle. Right, but now we're separating our sister chromatids. So it's going to look like this. Right, so the chromosomes that are on the left are being pulled to the left. The chromatids on the right are being pulled to the right. You know, here's where we lined up in metaphase. So you can see how we're pulling sets of chromosomes, one to each side. Right, and then when we get to telophase, what's going to happen with this is we're going to see this kind of little indentation show up like this. Right, our spindle is breaking down, so we're not going to have the spindle anymore. Okay, what we're going to have are chromosomes that we're pulling to each side. Right, so our chromosomes look like this that we pulled to each side. And we're starting to reform nuclear membranes around each of those sets. All right, so that is mitosis. Right, the goal: separate chromosomes. Right, now, I'm not showing you two new cells. This is still one cell. It looks a little bit funky, but that cell in telophase is still a single cell that we're developing two different nuclei in. Right, so that's our set of stuff through mitosis. So remember we had this up here. These things are interphase. Right, so mitosis is technically all this stuff. Right, that's the steps that we saw with mitosis. 5 steps, right? Here's 1, 2, 3, 4, 5. Right, if we continued that through cytokinesis, then we would split it and have our two separate, they're called daughter cells. Right, so let me show you a little different way of looking at this. So if we had our cell at the beginning, it's called a parent cell. Right, and let's say this parent cell is 2N. And we go through this cell cycle and we're going to create these two cells that are called daughter cells. Hey, these daughter cells would still be 2N. Hey, that's what we're trying to do with mitosis. We actually want the chromosome numbers and all that stuff to be the same. Right, so to do this, we would have to do interphase, then mitosis, then cytokinesis. Right, we've got to do all 3 of those things to get from our one parent cell to our 2 daughter cells. Hey, and then let me show you just one last thing with this, because I showed you if the cell we're starting with is 2N, then what we're ending with is 2N. The daughter cells are 2N. I mean, we may actually see numbers for this too, right? Like if we said this is 2N is 46, then this is going to be 2N is 46, and this is going to be 2N is 46. like all those are going to be the same. And we can do that, the number thing, even without any of the other detail stuff, right? So we see this picture. If you see this picture and we say this is a parent cell, these are daughter cells, you got to be thinking all this stuff is happening, right? And no matter what the numbers are, we could do this. So if this says 2n is 10, this is going to be 2n is 10, 2n is 10, right? Depends on the cell, the organism, etc., right? But we're maintaining that number. We're not changing that number with when we're using mitosis.